Hello, everyone. This is Herbert from Queen's University at Kingston, Canada. And here I'm introducing Overlay, the computational pipeline that we've been working on. And this pipeline takes the genome scale metabolic model, we're going to refer as M model, and the gene expression data under certain circumstances of the cell to investigate the detailed cellular metabolism. So the intuition with this study is simple. Like nowadays, measuring gene expression data, including RNA sequencing and like proteome, like profiling and things like that, are very easy and very accurate under various experimental and like various environmental conditions. However, like metabolism is still hard to measure, and it's like even if we somehow measure it, it's still very noisy. But it's metabolism is important in say biotechnology applications or like biochemical and infectious disease. So here we have a strong motivation in finding a method to predict the metabolism from given gene expression data. So and the tool we're going to use is genome scale metabolic modeling. And yeah, this overlay workflow is highly automated, so it's easy to use. And here's the workflow of this study, and the red two columns are actually the workflow of overlay pipeline. So we're working on a case study, basically for the sake of this pipeline development. We are investigating the tag accumulation, triacylglycerol accumulation in chlamydomonas green-hardy, a microalgae. So we're using SM model, and we did some like model refinement. And then like from that M model, we built a protein constrained M model called PC model, also referred as EC model in some other literatures, but basically it contains information of proteins and enzymes. In the model, it's not only it's not only metabolic, and then from that we can get really the protein constrained optimal solution, optimal metabolism. That's without like any data, any expression data. And here we can do the data interpretation using a two-step quadratic optimization algorithm, and we can basically arrive at a context-specific metabolism or context-specific model that referring to a context-specific metabolism under the cases where this data is taken. And we can suggest engineering strategy for boosting the triacylglycerol production. So here's basically the result of the what we call the protein or weird optimal metabolism. It's a diamond here. So I mean the optimal metabolism can be predicted by M model only, but protein constrained model, the PC model predicts better because we add in the we account for the proteome constraint and enzymatic rate constant, of course. So the basically the constraint is more complete, and we can see the autotrophic, mixotrophic, and heterotrophic metabolism for chlamydomonas green-hardy, and yeah, and clearly like you can see in detail later, but basically the autotrophic has more chloroplast involvement and larger photosystem than the heterotrophic. And mixotrophic is even more interesting, and we recommend mixotrophic conditions for biotechnology applications. And on the in the convex QP, non-convex and convex QP, we call it two-step QP. And we can see like how well the model is interpreting the data, or how well the model is uptaking the data, because we are doing optimization that minimizing the difference between the experimentally measured expression versus the protein in the model. So and the non-convex QP reduces this difference by adjusting the enzymatic rate constant. We call it K effective. So yeah, and as you can see from the histogram here, we're not really adjusting too much. We're adjusting really a small portion, and it achieves better like fitting, which will allow better downstream analysis. And as you can probably appreciate, if we have very comprehensive data and very good computational power, the enzymatic rate constant estimation will be quite good for any cell. And then there's basically the main result of what we call the context-specific metabolism or flux omics. That is the flux. That is the flux omics basic estimation on the cell where the gene expression measurements are taken. So any suggestion we can come out of this can be really applied to the lab rather than being very generic and vague and difficult to apply, which is a weakness basically inherent by the modeling. So we can see like here we have the tag production, which is right here. The bar is basically the flux variability. So yeah, so we can see the flux variability, the maximum tag production, like it's actually over time reduced and then increased over the course of the experiment. And there are certain reactions that and the black line is the expression level. So we can see the like for some flux, for some certain reactions, their flux is highly correlated with the expression. We call it expression dependent reactions. And there are other reactions that are like almost nothing to do, like such as this one. And this is the acid kinase. And yeah, there are many, and that's the ATP, that's the mitochondrial ATP synthesis complex 5. So that's, you can say it's almost nothing to do. And from this, like if you want to arrive at the engineering strategy sooner, we can actually look at what flux omic is almost identical or is affecting the objective function, and in this case is the fatty acid synthesis. And we can see the 4-step fatty acid synthesis almost have identical flux omics to the final result. And in turn, we can see their expression, the fatty acid synthesis steps expression are highly correlated with this flux omics. So in this case, we know that it's this reaction that's dictating the fatty acid synthesis rate, which in turn dictates the tag production rate. Well, given our estimated rate constant, so like it's also possible that some other reactions are dictating the rate of tag production as well, like maybe like somewhere along the line of tag synthesis, and the relationship of these reactions you can see in this graph right here. So this is a very rough introduction, and yeah, if you're interested, you're extremely welcome to talk to me directly, and our overlay workflow is posted, as well as our project and the paper's preprint. And this work is supported by these organizations, and we are in QSCSB, Queen's University System Biology Group. Thank you so much, and looking forward to talk to you soon.